All right, so you've got the mechanics of your microscope down from our last lesson. You know exactly how those lenses bend light and how the stage operates. Now we're going to use this explainer to unlock the invisible, clinical mystery sitting right there on your glass slide. For anyone heading into a healthcare career, this is literally where the rubber meets the road. We're shifting gears from the physics of the equipment to the chemistry of your sample, so you can actually see the pathogens you're trying to fight. Here's a quick look at our roadmap for today. We'll be covering preparing the specimen, the chemistry of color, staining strategies, the gram stain protocol, and we'll wrap up with the clinical value. Let's jump right into Section 1, preparing the specimen, fresh versus fixed. Because before we can even think about adding those brilliant colors, we have to prep our biological canvas. Think about this for a second. How do we actually bridge that gap between simply shining a beam of light through a glass lens and actively tracking the dynamic, microscopic properties of a dangerous pathogen? You see, most biological cells are essentially just transparent bags of water. To actually see them, we have to manipulate them. And we primarily do this in two distinct ways. First up, we've got fresh preparations, commonly known as wet mounts. This is where you suspend your sample in a tiny liquid droplet right beneath a cover slip. You're going to use this when you need to observe direct, real-time, living organisms. If you want to track true biological motility or see how a cell naturally groups up without disturbing it, a wet mount is your go-to. On the flip side, we have fixed preparations, or smears. For these, you spread a liquid film of cells across a slide, let it air dry completely, and then run it through a localized heat source or a chemical bath. This step is absolutely crucial. Heating it literally bakes the cellular proteins permanently onto the glass. It immobilizes the cells and preps that cell matrix to endure the heavy washing of the multi-step staining protocols we're about to discuss. That brings us to Section 2, the chemistry of color. Let's look at the specific biological dyes that make these clear, heat-fixed cells completely visible to the human eye. Now, what's really fascinating here is understanding that a biological dye isn't just a simple color, like the paint you'd slap on a wall. It's actually a highly targeted electrochemical tool. Scientifically speaking, dyes are organic chemical compounds containing something called a chromophore. That chromophore is special because it carries a distinct electrical charge. And that exact charge is the secret sauce to creating contrast against a bright microscope background. Which brings us to a fundamental electrochemical metaphor you absolutely need to remember. At a neutral pH, most bacterial cells are covered in negative phosphate and carboxyl groups on their outer layers. Because of this, bacteria act exactly like negatively charged magnets. So think about the basic or cationic dyes. Their chromophores have a positive charge, and since opposites attract, these dyes actively bind directly to those negatively charged cellular structures. Conversely, you have acidic or anionic dyes, which carry a negative charge. Two negatives repel each other, right? So the dye is pushed away from the cell, leaving the actual cell body totally clear while it stains the background instead. Moving on to Section 3, staining strategies, positive versus negative. We take those positively and negatively charged dyes we just talked about, and we use them to build two entirely different staining architectures. Positive staining employs those positively charged basic dyes, things like crystal violet or methylene blue. The dye is electrostatically drawn right into the cell, coloring the organism directly while leaving the surrounding background totally clear. Negative staining does the exact opposite. By using negatively charged acidic dyes like India ink, the dye is actively repelled by the cell's surface. This creates a really dark background, making the cell stand out as a clear, glowing silhouette. And there's a massive clinical advantage to negative staining. It completely avoids heat fixation, which means absolutely no cell shrinkage occurs. You are seeing the pathogen's true unshrunken size. This perfectly illustrates our overall hierarchy of stains. Starting at the top, simple stains use just a single dye to give you rapid, high-level details. Is it a round coccus or a long rod? Simple as that. Moving down, differential stains use multiple steps, usually a primary stain, a selective decolorizer, and a counterstain, to categorize complex mixtures of bacteria based entirely on their wall chemistry. Finally, structural stains are highly specialized protocols used to map out specific clinical defenses. We're talking about things like slippery outer capsules, or the microscopic flagellar filaments a bacteria uses to swim around. All right, Section 4, the gram stain protocol. Listen up, because this is the absolute climax of clinical microscopy and arguably the most vital differential stain you will ever perform in healthcare. Let's walk through this four-step chemical mechanism together. Step 1, your primary stain. Crystal violet turns every single cell on that slide a deep purple.
Step 2, Graham's iodine is added as a mordant, forming massive crystal violet iodine complexes that get trapped inside the cell walls. Step 3 is the magic moment, decolorization with a volatile solvent, like ethanol. For bacteria with thin walls and lipid-rich membranes, the alcohol dissolves the lipids and washes that purple color completely out. But for bacteria with thick peptidoglycan walls, the alcohol shrinks the wall, trapping the purple dye securely inside. Finally, step 4, the counterstain. You apply safranin. The cells that were cleared in step 3 soak it up and turn pink, while the thick-walled purple cells remain unaffected.
So the crucial takeaway for you, and I mean absolutely essential for your exams and your future careers, is memorizing this dichotomy. This is your golden rule. Most clinically significant pathogenic bacteria fall into one of these two camps. Gram-positive bacteria have that incredibly thick peptidoglycan wall, they trap the complex, they stain purple. Gram-negative bacteria have a much thinner wall and a lipid membrane that gets dissolved by the alcohol. They wash clear, and then they take up the pink safranin counterstain. They stain pink or red. Positive is purple, negative is pink. Master that, because as you'll see, this color difference means everything. Which brings us perfectly to our final section, clinical value. Let's step out of the chemistry lab for a second and walk right into the hospital to see exactly why this purple and pink difference saves patient lives every single day. Picture this. A critically ill patient presents in the ER with a severe, unknown infection. Doctors do not have days to sit around waiting for deep genetic lab work to come back. But by simply swapping the patient, making a quick smear, and performing a rapid gram stain, seeing purple or pink under that microscope allows a physician to instantly deduce the pathogen's cell wall morphology. That knowledge alone dictates whether they should target the infection with narrow-spectrum antibiotics or if they need to drop the hammer with broad-spectrum antibiotics right then and there. I actually want to read this directly from the source material to really underscore its importance. The gram stain, quote, rapidly categorizes an unknown bacterial specimen into gram-positive purple or gram-negative pink lineages, allowing targeted, life-saving antibiotics. Mastering this relatively simple protocol of specimen preparation directly dictates the success of patient treatment. You aren't just making a slide look pretty, you are literally arming the physician with the exact intelligence they need to save a life. And that leaves us with this final thought. You now know how to physically manage the microscope, and you know the fundamental chemistry of making the invisible visible, and, crucially, whether those pathogens are gram-positive purple or gram-negative pink. So as you move forward into your next explainer on the advanced 6 I's of microbiology, I want you to ask yourself, how will you utilize these vibrant, targeted staining strategies to skillfully inoculate, isolate, and ultimately identify the unknown pathogens you're going to encounter in your clinical career? Keep that question front and center as we continue to explore this incredible microbial world.